So we have seen this is the bone pattern of the limbs, the stylopod, the zeugopod, and the otopod. But limbs, they grow according to the three-dimensional structure. So it means that when they grow according to three axes. This is the anterior posterior axis of the limb, or anterior posterior. Okay, so the anterior posterior, it means that our little finger or the toe mark the posterior side, and the thumb will be at the anterior side. The second axis of growth is called proximal distal. So proximal distal, it means that during the process of ossification of the cartilage to form bones, usually the limb, okay, or the cells that will differentiate first from cartilage to bone, they will form first the stylopod, which are at the most proximal side. Okay, it means near the point of attachment of the limb to the body. Okay, for example, here we are taking the example of the hand. Okay, this is the proximal and this is the distal. Okay, the most far away is the distal, the near is the proximal. Okay, so it means that the stylopod is the most proximal, then we have the zeugopod, and the most distal is the otopod. So we have the anterior posterior axis of growth, the proximal distal axis of growth, and we have the dorsal ventral. This is the dorsal ventral axis of growth. It means if you look here at this hand, for example, the palm, okay, the palms are ventral. Okay, palms, it means in Arabic راحة الكف يعني. Okay, so this part is ventral. Okay, while at the opposite side, if we turn our hand the other way around, okay, we have the dorsal side where the knuckles or the articulation between bones are or appear, or they are present. Okay, so we have this kind of three-dimensional structure of limbs. And of course each axis is regulated by a specific pathway. That's why we talk about molecular regulation of limb formation. For example, the proximal distal axis is controlled by the fibroblast growth family, the FGF. Okay, we have seen before in the last chapter, or in the chapter 3, I think. Yes, we have seen the FGF signaling pathway, if you remember. Okay, this controls the proximal distal axis of growth, and we talked about this function within the functions of the FGF. Is that it's important for the growth of the limbs according to the proximal distal axis. Okay, so the second pathway is the sonic hedgehog. It controls the anterior posterior axis. Okay, the thumb, pinky is the small finger. The dorsal ventral axis, it means the knuckle, dorsally the palm is ventral, is regulated by Wnt7a. Okay, so we have for each axis we have a specific signaling pathway to regulate it, and you should know here that these pathways are not independent. So during the growth we have interactions between these different proteins, between FGF, sonic hedgehog, and Wnt7a. We have a mutual or reciprocal interactions, so one can support the another one in order to determine the differentiation of the different cell types according to the three axes, in order to organize and coordinate the growth of the limbs according to these three different axes. So here I'm showing you the formation of the forelimb, for example, and any other limb, it will go the same way. For example, the formation of the forelimb in salamander, okay, which is a species very close to the frogs or the xenopus. So you see that during this tail bud stage, this is the bud of the forelimb. It's a kind of disc formed laterally from each side. Okay, we have 2 forelimbs and 2 hind limbs. So each disc is formed at the center by a region called free limb. This region will give the limb itself. Okay, but this free limb is flanked from each side by cells, for example, here called peribrachial flank tissue. This tissue is a region made of mesoderm surrounding the free limb, and this region is known to compensate the loss of any cell present in the free limb, because this region is highly important for the formation of the limb, and any cell death here will be compensated by cells coming from the peribrachial flank tissue that can replace them. So from the other side, the free limb is also surrounded by the shoulder girdle, okay, which are bones that connect this upper limb with the axial skeleton, okay, with the axial skeleton of the body. It's like, for example, in human, like we have the clavicle and the scapula, these bones that connect the upper limbs, for example, with the axial skeleton. Here it's the same. We have this shoulder girdle that will give the bones that can attach the upper limbs with the axial skeleton. So when we say limb field, for example, in this organism, limb field, it means all the cells involved in limb formation, and here I mean the free limb, the peribrachial, and the shoulder girdle. So the limb field is known to be a harmonious equipotential system. What does it mean? It means all the cells forming the limb fields have or are equipotential. What does it mean? Equipotential, it means they have similar it means equivalent potential, similar potential to generate any missing part of the limb. So the limb field has the ability to regenerate its missing part according to different experience done. I will explain you, for example, one of those experiences. So in the tail bud stage, any half of the limb disc is able to regenerate the entire limb when grafted to a new site. For example, if we take just a part of it, okay, and we grafted it somewhere else in another organism, this part of the limb disc is able to generate the entire limb when grafted to this new site. So here in this experiment, they took a limb disc and they splitted it in a vertical way, okay? So they splitted it in a vertical way like this, just to cut it into different pieces, and they put between these different pieces barriers just to prevent the reassembly of these different segments. Here they cut it vertically into 2 or 3 segments, for example. After some time, of course in vitro they are working, they found out that each segment of those could develop into a full limb, okay? And from this experiment they have concluded that the limb field is a harmonious equipotential system, because each part or each segment is able to regenerate all the missing parts. And they have seen that this is the formation of limbs is a equipotential system not only in frogs or salamanders, but also in the sea urchin, okay? So the limb field is also a harmonious equipotential system, since each cell can be instructed to form any part of the limb, okay? So it's the same property in these organisms, that the limb field is an equipotential or harmonious equipotential system. And here I'm showing you a multi-legged frog. So as you can see here, this is the skeleton of this multi-legged frog. We can see that it's multi-legged because usually we have 2 forelegs or 4 forearms or 4 limbs and 2 hind limbs. But here we have at the left side 2 hind limbs from one side and like 1, 2 and 3 from the right side and 2 from the left side. Okay, so it means that we have extra legs. How this could happen? So in one lab working on frogs, okay, they realized that they started having this kind of phenotype, obtaining multi-legged frogs. So they thought that they had an important mutation maybe that induced the formation of this kind of phenotype, but this was not the case. Actually, in this lab was close to another lab working on nematoda, okay, the parasitic trematoda worms, and some of these worms escaped from this lab to the next lab working on frogs, and these worms infected the abdominal part of the larvae in these frogs, okay, and they splitted the limb bud into different fragments, okay. So these worms laid eggs, okay, and these eggs splitted the tadpole limb, okay, into different fragments, okay, and each fragment of the limb bud developed into a full limb. That's why we got multiple limbs. For example, from the left side here, each limb field, let's say, has been split into 2 parts. We got 2 forelegs. Here each limb has been split into 3 parts. Each part was able to regenerate the missing parts and to form one full limb, okay. So, and this is another confirmation about the potential or about the property of the limb field as a harmonious equipotential system and its ability to regenerate the missing part when it's splitted.